Yeah, yes, I think it seems to work now. So I can hear myself as well. Hi, everyone. Welcome to another Saturday evening live stream. I say Saturday evening because evening where I am, I'm living at. But I know that many of you are all around the world. Sometimes it's maybe in your place it's in the morning or quite late at night or even in the middle of the night. Today I want to do a little bit of crystal microscopy again, but not only. As you've seen already on the thumbnail, I want to put some urine under the microscope and grow some urea crystals. A little bit of a backstory because some of you might think that this is a little bit of an unusual specimen. Well, it was actually yesterday when I was teaching biology. We were just talking about the kidneys and urine production. Then actually one of my students asked me if I can put urine under the microscope, and my first instinct was, well, actually it's not a very interesting specimen. Actually, urine is kind of boring unless, and then that's what I remembered, unless you grow crystals. And this is what I decided to do. Let's grow a couple of crystals from urine. And yeah, if you're new here, then I just want to inform you there's a chat. And if you want to ask some questions, then please indicate my name at Microbehunter so that it's easier for me to find the comments that are directed to me. What else do I want to do today? I would like to also put the exoskeleton of Spiky on a permanently mounted slide. What in the world am I talking about? Well, it's a little carpet beetle which was molting, and last week there was a suggestion if I cannot make a permanent mount of the molted exoskeleton, I'll try this as well. And depending on how much time we have, I'd like to look at this terrarium that I made, and it looks pretty sad right now. I made this several months ago, also during a live stream, and I would like to have a look at how this one actually developed. Okay, so that's kind of a little bit the plan, but as every week I'm a little bit flexible with the time and with the topic, so if you have any specific requests, of course I can also try to include those. Okay, so what I'm going to do is I'm going to cut the greeting short. I would like to say hello all around the world, as always, and I'm going to first show you before I actually start making the crystals, I just want to show you the prepared slide, which I already prepared before this live stream. Over here, you can actually see this. These are some crystals, possibly urea, possibly sodium chloride, because it's also in urine, possibly some phosphates. However, I have to tell you, I have already made slides from urine before, which look actually nicer than this. But all of those things that you see here, these are crystals. So I'm going to also show you a couple of pictures that I made. One of the things that I quickly realized is that they're different. The crystals have different shapes, right? And this is actually a sign that, yeah, we're probably looking at a mix of different salts in the urine here. Obviously not just urea, which is the waste product, but also evidently some other minerals as well. Yeah, I'm going to show you how I've prepared that. I'm also going to show you then some pure urea, which is actually quite nice looking as well. Okay. Every now and then I do interrupt myself a little bit because I'm also reading the chat, but I would like to respond to the off-topic questions, which there might be, which is fine, towards the end of this live stream. Okay, so I'm just scanning around here. So a little bit some additional info here. Look what I've got here on my table. Okay, so I would like to, yeah, that's basically how urea looks like. It's an organic substance. It was actually the first organic substance that was actually made artificially. And I did a little bit of research here because the urea is not only a waste product in our body, but it's actually one of the most produced chemicals worldwide. 200 million tons of urea are produced. Look, I even have some pure urea here. Yeah, and I'm going to be also using this and putting this under the microscope. It's a little bit too bright, but these look like little pellets that we have here. That is pure urea. Okay, and yeah, it's non-toxic. That's pretty cool. It's used for making glues, resins, fertilizer, a lot of fertilizer, even for caffeine. I didn't know that. You use urea for making caffeine. And yeah, in 1797, this guy over here, I don't even want to pronounce his name, it's so difficult. He was actually the first one who discovered that urea urine can crystallize, yeah, and it forms urea crystals. And in 1828, Friedrich Wöhler, he was the first one to actually artificially make urea, right? Yeah, the thing that you've all been waiting for we're not just going to put this here under the microscope, but look what I have here. Guess what this is? Yeah, that's going to be our sample. Okay, and I'm also going to we're going to put also some fresh urine under the microscope just to show you what's there. What else do we need? If we want to grow the crystals, we need, I have a hot plate here. Yeah, you can adjust the temperature here, 80 degrees. This is a coffee warmer, which has already been active quite a bit. I'm now going to start growing some of these crystals that you've just seen, okay? So, what do you need? Of course, a microscope slide. And what I'm going to do now, however, is because I just recently washed those slides, I'm going to clean them again, just to make sure that they're really clean. Because I want to have very nice crystals, and if there's any dirt on here, then every dirt can act as a crystallization seed. Okay, and I place it directly on here, obviously, right? And the hot plate allows you, speeds up the evaporation process. I will take a microliter pipette. And now, folks, we gotta be a little bit patient, okay? So I hope you don't mind, but this is gonna take now a couple of minutes, right? So let me put this over here. Yeah. What does pure urea smell like? This one here, it smells like nothing, okay? Really, it's interesting. No, it's totally neutral. Yeah. Yeah. By the way, where do I have the urea from? I got this. I'm also from one fellow microscopist in Germany who sent me a couple of several months ago a whole pack of different crystals to put under the microscope. So what I'm going to do now is I'm going to put it on here. Okay. So I want to kind of make sure that the drop is fairly small, and I allow to evaporate it, and I will add another drop. And the reason is the following: depending on how much you've been drinking water before, and how much salt you've eaten before, the urine will contain different concentrations of urea and salts. So I've made already specimens, and they just put one drop on here and had plenty of crystals. But this one over here does not produce as many crystals, so I have to actually add more. Okay, simply to increase the density. And the concentration rather of the salts and of the urea, yeah. No, it doesn't. I've even dissolved some of the question. Curious if it smells something after it's been dissolved. No, it's totally neutral. This urea is yeah. The ammonia. Well, there is actually no ammonia. Luckily. Unless it maybe dissociates and forms ammonia, but actually pure urea is it should not contain ammonia, because ammonia, also in urine, it is actually quite toxic. Yeah, that's the reason. For example, fish are able to produce their nitrogen waste in the form of ammonia, and it's given off over the gills. But in humans, this would be actually a problem. Yeah. Yeah, so I've added now, I don't know, 2 or 3 of these drops here. You can see that it starts to turn a little bit cloudy. I can imagine maybe this is a little bit, the white stuff could already be maybe something precipitated, or maybe some tiny crystals that are already forming. Yeah, yeah, that's a little bit something that requires patience. The crystals will look very different. When I put some pure urea under the microscope, you will see it will look totally different. And this has to do, of course, also not only with the concentration, but also certain impurities which might be present. Okay, you know what? I'm just going to leave it as it is here. I'm going to allow this to dry, and let's see how nice these crystals are. Okay. I remember when I got my first microscope, when I was 6 years old, for Christmas, a toy microscope. The first thing that I put under the microscope was some of my urine, and I was extremely disappointed because I could not see anything. So I thought I did something wrong, but indeed, pure urine actually is pretty free of bacteria. Should be. Yeah, it actually should be fairly clean. If there is some stuff in there, like bacteria, so then you've got an infection, which should not be the case. So this is yeah, something. This is hot. I don't know. Now I get a little bit the impression that this might be a little bit too concentrated. I just realized. Oh, okay. I just realized this might be a little bit too concentrated. This is way too thick. So what I'm going to do now is the following. I'm going to add another one, but I'm going to spread it out a little bit. Okay. I'm going to spread it out a little bit here to lower, yeah, to make it a little bit less concentrated there. Okay. Yeah, so that is, yes, thank you for the comment. It is on topic. I love crystal microscopy. I would be interested to know all samples your friend sent you, maybe a topic for later. Yes, thank you for the recommendation. However, a few years ago, 2 years ago maybe, I actually did put all of them under the microscope, but many of you might not have seen that stream. It might have, I think, even been on my other channel. So I will do that again. I will pick up on that again, and I will do this, yeah. I read some of the other comments here. Okay, confocal and parfocal versions. That's an interesting. I don't know what do you mean with confocal. Parfocal actually means parfocal means that the it's about stereo microscopes. Parfocal means that the image that you actually get through the eyepieces is the same focus as the ones on through the camera. This means parfocality. Yeah. So. Here it is. Let's give this one here a try. I'm going to turn this off. It's still pretty hot, so I have to cool it all down a little bit. I'll put it on a cold. I'll put it on a cold slide here, simply to cool it off a little bit. Yeah, it's getting okay. Okay, so let's put this away. Okay, so just checking if it's. So I'm going to now do the following. I'm going to switch over to the microscope view again. So that's still the slide from before. Let's take this away. And let's put this one on here. Yeah, I need to go down a little bit with the. Yep, and this is at low magnification, and we can already see tiny crystals. This looks a little bit messy. But over here, maybe. So I'm using polarized light now. Okay. Yeah, here, look, the crystals here on the side, they look again a little different. They look almost a little bit needle, elongated and needle-shaped. And this actually is indeed a sign if you have different crystals that essentially we're not talking about a pure solution, but actually one that contains a mix of different substances. Yeah. So I'm because confocal, there is something called confocal microscopy. I'm reading the which is actually called laser confocal microscopy, and maybe it has a different meaning here. Yeah. So this is so there has been another on-topic request about putting it under regular polarization filters. I will tell you the problem. I will try to do that, but I will tell you the problem with that. So first of all, background info. I'm using polarized light right now. However, it is also DIC microscopy. So I have I'm able to change the colors here. But this is not the normal way of polarized light microscopy. If you want to use cross-polarized filters, then that's also possible. However, I have a problem that the crystals are so small that I have to use a very high magnification, and then I don't have enough space to put a polarizing filter between the slide and the objective. Yeah, so it's a space problem that I have. So what I'm going to try to do is I'm going to try to put it on my stereo microscope, and we're going to see what we're able to see there. Okay, if you want to see it without polarization, okay, just regular bright field. This is how it looks like. Okay, I know it's not quite as colorful, but you're still able to see the crystal. So without any polarization, without anything, this is how you're going to see it. So you can still see the crystals. Yeah. I want to show you something else, what I've made, not using this microscope, but using a standard bright field microscope with oblique illumination. I'm going to show you a little slideshow in a minute or so, okay? Okay, so you see that here the crystal, that's regular bright field. I'm now going to show you some really beautiful crystals that I made a few years ago. However, I was not able to reproduce it anymore. So trial and error. So here is the slideshow. Okay, look at these crystals. Basically, I use the same technique. And this looks really nice. And I've used oblique illumination. So basically, I used a filter, a dark field filter, and made it a little bit off center, and it gives this three-dimensional impression. Honestly, I think this looks actually nicer than the one that I just showed you using my microscope right now. Yeah. So so these flower-like patterns. Yeah. Here again, two different crystals. I was really lucky that I got just the concentration right to see those. So if you've got a standard microscope with a filter holder at home, nothing fancy, not even polarization, then yeah, it's possible to see it like that. Yeah, exactly. It's almost like scanning electron microscope. Yeah. So just wanted to share this with you, that even with very simple techniques, it's quite nicely possible to get quite beautiful results here. So yeah, let's go back a little bit to the to this one over here. Let's add a little bit of color again. I have to turn it up. Yeah, and let's have a look over here where it's a little bit, I don't know. You see over here the crystals here, you're barely able to see them. They're very tiny. Yeah? That's why I added more of this of the urine simply to increase the concentration a little bit. Yeah, I think you get the idea. What I'm gonna do now is the following. Before I show you the pure urea, I want to put some liquid urine under the microscope, and I'm going to stain it, because there is also lots of other stuff, of course, in urine. And if you add a little stain, then you can actually see much better what's floating around in there. Yeah. So every now and then when I make a short So the pictures, Tini Coco asks, is that oblique? The one that I showed you before the slideshow? This one here, yeah, that's oblique illumination. So no polarization, just a regular dark field patch stop filter, which I offset a little bit, basically decentered it, and then it shines light from the side, and then it gives this three-dimensional appearance. And I have to admit, I was quite surprised that the result was that good. So, okay, let's do the following. Let's not make crystals, but let's put some urine under the microscope, some pure urine. And the interesting thing is that let's go back to the desk view here. If you look at the urine a little bit against the light, you're gonna see it is a little bit cloudy. There is stuff floating around in there. Yeah, you won't be able to see it here. And it's usually like this, that of course there are cells from the body breaking off and also being washed out. So it's urine is while generally mostly pretty clean, because there shouldn't be any bacteria growing in there unless you have a medical problem. Yeah, you basically also have some other stuff floating around. So, and the thing, however, is the following, that it might be difficult to see. And for this reason, I'm going to now throw this away. I'm going to add a small amount of stain, and simply to illustrate that what's in there. I'm just going to use now crystal violet. You can also use methylene blue or whatever stain you want, but I found that crystal violet works quite well. And always be super careful that I don't spill this. Don't tip it over. And so I have to go like this. Take a tiny amount. Immediately close it again. I mean, I don't know. I think this might be even a little bit too much. Let's mix it. Okay. And Throw it away, and we add a cover glass, of course. So this is again, this is now stained urine, simply to make the things that are floating around in there a little bit better visible. Okay, so that's the idea. So, and if we see anything that's kind of stained blue, see, it's too much. We need to make sure that the cover glass is not floating around so much. Okay. Urine only becomes a hygiene, or should mainly becomes a hygienic problem if it's standing around for some time and if actually bacteria start to reproduce in it. Yeah. So, because of course all of this organic stuff that's in there are good nutrients. So let's put this one here back into the microscope. So, and let's see what we are able to find here. It's a little too, you can already see there is lots of stuff. All of the things that you see here, right? Yeah, was kind of stuff that was in the urine. I can say maybe cell fragments, maybe, I don't know. Yeah, so it is actually not just a solution, but actually a little bit of a suspension as well. Let me take the thing away, the filter away. Yeah. So just to illustrate, yeah, that there is lots of stuff in urine, yeah, that is stained. Yeah, you see there's some stuff still flowing around. So this was basically simply an illustration. Of course, you're not able to see all of these things if you don't stain it. Yeah. So I think once I remember some time ago, I was actually lucky enough to actually find also some epithelial cells yeah, from the lining in there. Yeah, just wanted to illustrate this. This is a bubble here. Just wanted to show this to you a little bit. Yeah. The question is the following: is what are these little bubbles? Indeed. It's an interesting question. I'm also not quite sure. It almost looks like if these bubbles are very round, those tiny ones, almost like tiny fat droplets, but I can hardly imagine this. Yeah. It could be dead. I mean, maybe at this magnification, these are not dead red blood cells because they would be much larger. Okay. If there were any blood cells in there, then they should be larger. What I can, however, do, and I have not tried that yet, but again, that might be again something for an upcoming live stream, is to actually put it under the centrifuge. Okay? Because I do see there is some stuff in there, and if you actually spin it and put it under the centrifuge, maybe you're able to, few cells that are actually in there, maybe you're able to actually concentrate them. But I just wanted to show this to you actually simply to illustrate that there is plenty of stuff in urine as well. Okay, and look at this here. I don't know, little weird stuff. Could it be? Look, I've seen this also before. Those spiky things here. I have the suspicion that something is starting to crystallize out here as well. Look at these weird, yeah. I've seen this also before, and I think this might also be some crystals that are actually starting to grow. Because these look kind of, yeah, weird. It could be maybe that there is also a reaction with the stain, and that maybe some things start to precipitate out. Yeah, I know, pea spiders. If they were really, but you see, they were not here before, right? And now they start to appear. So I think I do suppose that these are also crystals. Yeah. So, and they seem to become a little bit bigger. And I cannot go up with the magnification. Actually, I'm already at 60 times. That's the question. What color do they have? I mean, they do. They look a little bit dark blue, dark pink. Right now I'm not using any yeah, any filters or so. So this is actually bright field. Yeah. Okay, I think yeah. Would be interesting to see if they actually go here they are again. Yeah, plenty of stuff on here. So what I'm going to do now is the following: is I'm going to put some pure urea now under the microscope, because we do want to see something beautiful as well, right? We do also want to see something slightly artistic as well. So I'm going to go over here to the desk view, and I'm going to put the slides away. I need a little bit of water here. I mean, I already prepared something here, but I'm gonna do this again. So a little bit more water, and I will take one of these. Yeah, it's urea. That's German. Harnstoff. That's German for urea. They sell them in those pellets. Yeah, and I'll just take one of them here, and I put it here into the water. Okay, and wait until it dissolves. We're just gonna take 3 minutes, 4 minutes maybe. Okay. And then we're gonna do the same thing again. Going to put it on a slide, and I'm going to evaporate the water on the hot plate, and then we're hopefully able to see some more beautiful crystals. And in the meantime, I'm going to read again some of the comments. To the red dots. Would you take the cover glass off? Okay. Okay. I get it. You want to basically see. Let's see this here. We want to see what happens actually to those weird spider-looking like structures that we occasionally see here. Okay. And if I take off the cover glass, then you know what I'm gonna do? I think I'm gonna leave, and then we're gonna see what happens later. But I think I might actually do is I might leave it on because it's already starting to dry. And if I put it on the warm plate, then it's going to probably dry anyway. But if I take it off now, maybe I'm going to destroy maybe the structure a little bit. So what I'm going to do is I'm going to leave it on here, and we're going to keep on evaporating this directly beneath the cover glass, I would suggest. Okay, so you see over here that it's already starting to become smaller and smaller. Okay, and yeah, I think that's gonna be enough. A new pipette tip, and I'm going to take this here, goes on here, and I'm going to use a little bit more, but this one I'm going to spread over the slide. Yeah, you see, it depends a little bit on. Yeah, sometimes the surface tension is so big that. So, and let's do the following. Let's put it again on the hot plate. And let's wait a minute or so. I mean, in case you wonder, I did prepare, of course, before the start of the live stream, a backup in case something goes wrong. Yeah. Okay, there is a discussion about the. Okay, somebody wants to know a little bit the German name of Urea Harnstoff. Okay, Harnstoff. Yeah. The word Harn is urine, and Stoff means substance. Yeah. So it's a compound word. Mm-hmm. Yeah. It's this because it was sent to me from a friend in Germany. He labeled all of these things in German. Huh? But the urea looks like pellets. Yeah, maybe we can focus a little bit better. Okay. Here, that's how it looks like. So Okay, let's. I'm reading a little bit the comments. Yes, there is a comment over here: to make bigger crystals, the fluid needs to crystallize over longer cooling time so they have time to grow. Indeed. And this I've also grown some of the crystals simply without a hot plate, simply allowing it to dry at room temperature. However, this does take quite a bit of time. Yeah. So. Yes. Okay. I mean, what we could do that. We could, or I could. You know what I could try to do is I could try to maybe set it at a very low temperature. I could set it at a very low temperature and then maybe let it dry over a course of half an hour while I show you some other stuff. Okay. Yeah. Okay. Is it? So what's going on here? You see, it's interesting. I wonder if it's starting to crystallize. Maybe I'm going to touch it here a little bit. Ha! Did you just see this? That was a nice demonstration. Did you just see this? This was actually cool. This was it did not crystallize, right? And this was a little too fast, maybe. And when I touched it, then all of a sudden it started to crystallize all at once. So by touching it, I acted like a crystallization seed. Okay. Maybe the crystals are not so nice because, as we just said, for large crystals, it needs to crystallize slowly. Okay, but let's yeah. So let's have a look. In any case, I have to cool it a little bit. Yeah. Okay. So this was actually quite an unexpected demonstration. I have to cool it a little bit here. Okay. So this is gonna be fine. Where is the? Here it is. Yeah, of course. Yeah, this was a little too quick. I mean, it still kind of looks nice. Modern arts. Let's go down a little bit with the magnification here. Can play around a little bit with the colors. Yeah. So the question is, wasn't this contaminated with your touch? I touched it at one side, and this is the place where I touched it. Okay, I just see it over here. Here, right at the side, you can see that there are some scratches running across this, where it kind of streaked over it and scratched over it. So you can actually see that. And then from there, it started to form the crystals. Yeah, yeah, over the slide. Yeah. So beautiful patterns. This is now, this is now basically pure urea, and because it's pure, it's it also crystallizes a little bit in nicer patterns. What I'm going to do now is because you wanted to know, and I'm going to do this now in any case. I'm going to put it now directly under my stereo microscope under cross-polarized filters. Okay, so that we can also look at this there a little bit. So this one goes here. I'm going to take this off. I'm going to now use the stereo microscope view. I'm going to turn off. So let's put this on here. I think I need to get it a little bit into focus as well. Okay, I don't like to see the arrow. How to get the arrow away? The arrow goes away. I put the other filter over it. And I just drop it on it, okay, and then refocus. And this is basically, looks nice. This is basically now how it looks on the regular crossed polarized filters. Again, a little bit different. Yeah. Yeah. So let's have a look now at, so that's urea. Let's have now a look at how the urine looks like. I wonder if there's a difference. There you go. You see that over here? All of those dots, very tiny crystals. Again, maybe that's the reason. Maybe they crystallized a little too quickly. Let's zoom in. Yeah. Yeah, tiny crystals over here. Yeah. The nice thing about these is, of course, that they're easy to make. Salt is also quite easy, and some crystals are a little bit more difficult to make. For example, I found sugar, for example, more difficult because it doesn't dry quite as quickly. Okay, it likes to absorb also the moisture from the air. So what I'm gonna do now, desk view. Here we are again. Okay, I'm going to see if maybe at a low temperature, maybe briefly we can try to, I'm going to put those weird spider-like structures. Let's see if we can evaporate this here a little bit at low temperature, and then let's have a look later, how this looks like. What time is it? I think I'm slowly going to start, to move on, and I'm going to leave the. Where's the stereo microscope here? And let's put Spiky under the microscope again, because I wanted to mount the exoskeleton. And where is he here? When I checked everything, I said, where is he? He's gone. Okay, this is, by the way, the wrong light. You have to turn on the light from the top, right? And then I discovered that he's somehow hidden behind the thing that he molded. If you remember last week, I've added a few hair of myself and some fingernails. Everything's gone. Everything was eaten up. I think a couple of hair strands and some fingernails as a food source, and now indeed everything's gone. I did not open it up for a couple of days. Then I checked. See, now the whole thing is moving a little bit. So the difficulty is now, I don't know how to do that. I have to now make sure that I get a small part of this exoskeleton somehow on a microscope slide, so that I can make a permanent mount, because I never looked at those. And so I'm going to get myself a slide here. And this is going to be a little bit of a challenge with a lot of patience. And a scissor. Okay. So you see, Spiky is still moving, but it's hidden beneath. Yeah. So I thought he kind of escaped or something. Yeah. Look at this. I do get the impression that it starts moving. It starts moving every time when I shine the microscope light on it. So maybe he doesn't like the light. And because of all of the bristles, it's kind of difficult to separate them. This is an interesting question. What does he do for water? I've got this animal now in here in the petri dish since October. Have not given it any water. How does it actually get water? Well, what I do suppose is, like some other animals, maybe it's able to make its own water. If there's cell respiration happening, if in the cells, when organic material is oxidized to release energy, then water is a byproduct. So I can imagine that these animals are actually able to produce their own water due to cell respiration. I know that certain desert mammals are able to do that. They're able to make their own water when they're basically oxidizing the food that they eat. So for example, if you burn a candle, or anything for that matter, then you're not only producing carbon dioxide, but also water vapor. So, yeah. Now we see the spike is on the back. So, but you know what I'm gonna do is I'm going to somehow grab this thing here and I'm going to put it on here, and I have no idea now. I have to bend down a little bit. This is gonna be really difficult. And I'm going to try to get. See, it's a tiny. Just going to cut a little bit off here. Okay, a tiny part of this exoskeleton here on the slide. Let's put it back under the microscope to see if everything's intact. Here it is. Yeah, it's got to be somewhere. Let me check. I always have to check whether I lost it or not. This one over here is indeed just the exoskeleton. I was actually cutting it quite carefully. Where is he? That's interesting. Maybe this kind of shocked him so much. This could be that maybe because it got disturbed, it kind of started to fan out its bristles. Yeah, let's check back in a couple of minutes. So what I'm going to do now is I'm going to put the desk view back here. So this is the thing here. It's part of the exoskeleton here. I can also put that under the microscope over here. Let's check this. Okay, you see that it's pretty transparent. This specimen is known as the larva of a carpet beetle, and it's a pest. It's actually not a good sign to have it in your apartment because it damages carpets. It eats wool. Back in October, I found it crawling on my wall, and I immediately kept it because the bristles that it has are quite interesting. I'm a little bit it's the larva. So I'm a little bit, I think this might be a slightly difficult specimen here because it's a little bit round and it's not quite as flat. So what I'm going to do is I'm going to add now a little bit of mounting medium and a cover glass, and then hope that it's going to be pressed flat. So this is a little bit the thing. Why am I doing that? Well, that was a request last week. Okay, and I think it's quite an interesting thing because I myself never looked at it. So better not lose this. So here it is again. Now what? Look, I need to do the following. I'm going to put because I'll be working with a mounting medium, I'm going to put it on a sheet of paper. I need to find the center of the microscope slide so that I simply can make a nicer mount. So, and the next thing is I'm going to use this here to estimate. I'm going to make a cross in the center here to estimate where it's supposed to be. See, and then I can now move it more easily to the center. Okay, I don't know. Let's just use round cover glasses. Just see, there's a little bit of wind. I'm going to take one of those round cover glasses. I need to clean it off a little bit. Oh, that's not a problem. Where did I put the tissue paper? Here we go. Why do I not put it into alcohol first? That's a good question. No need to put it into alcohol because it is already dry. So I could put it into alcohol, but I wouldn't get any benefits. You put it normally into alcohol first in order to remove water. However, specimens like these, which are completely dry, don't have to be placed into alcohol. It doesn't hurt, but it's not needed. So let me put this somewhere where I can grab it more easily. Now the next thing is going to be a little bit of a, how do you say, requires patience. Actually, we put the drop first, small drop first. Then this goes in. Then we put a drop on top. So we have to make sure that it's properly here. This is a huge drop. Oh, this is probably too much. It doesn't matter. I don't care about beauty in this case. I just care about preservation, and so that I have a slide to look at also in the future. And the only thing that I worry about now is that it's reasonably centered. If there is too much mounting medium, it's just gonna take quite a bit longer to dry. It might not look very nice if it's too thick. There might be some mounting medium spilling out as well. And the mounting medium is already quite... So there might be also some air bubbles in there. And you know what? Doesn't matter. I only care that this is reasonably preserved. Okay. Yeah, there are still some air bubbles in here. Yeah, maybe they will disappear over time as the mounting medium dries. Who cares? I'm just going to leave it as it is. If you do this, you have to be a little bit careful that you do not get any mounting medium on the microscope objective. Yeah, so luckily there's not much spilling out here on the side, so I can actually do that. I'll put it under the microscope now, and let's have a look. Here it is, and here we go. Oh yeah, you know what? I'm going to take away the. Wow. Look at those. A few air bubbles. Huge air bubble over here. Who cares? Let's just... Yeah, but the bristles are remarkable. Look, there are even some of the legs visible over here. Yeah. And I was already happy to have seen, look at those defensive bristles that look like little arrows. Last week I talked about those. Yeah, those. Let me turn on the arrows now. Yeah, those weird looking ones. Yeah, I found them, quite a few of them in dust, in house dust. I always wondered, what are they? Until over a microscopy forum, somebody told me that these are actually the bristles of a carpet beetle. Yeah. Yeah. So this is quite nice. Again, this is not I would say, not the nicest preparation that I've ever made. It just there's lots of air bubbles in here. But it could quite well be that maybe in a couple of weeks or so that everything will become even yet a little bit brighter. You know what? I don't know. I'm going to try to carefully push down on here. Maybe I'm able to push the air bubbles out. A big air bubble being pushed out. Very nice. Of course, a lot of the mounting medium will also spill out probably on side of the cover glass. I hope that when I release it, that they're not going to be pulled it back in again. Don't be pulled back in. I need a vacuum chamber. Yes. Did I already tell you the story that I actually ordered a desiccator, a vacuum desiccator, several months ago, in order to do that, to attach a pump to, and then the desiccator arrived, and there was no possibility for me to connect the pump because they forgot the hole for the connector. Oh, gee, yeah. Okay. I mean, you get the idea. Yeah. Yeah, so exactly. There is now a lot of, as you can see also up there, there's a lot of mounting medium now which kind of spread out on the side. This can be removed afterwards. I'll, yeah, I don't know. I don't know if I'm gonna do that. I'm just gonna allow it to dry. Drying will now take several weeks. I mean, that's, yeah. Yes, there are the legs visible here. Yeah. So drying will take several weeks because it's a quite a slow curing, curing mounting medium. But this also gives it a lot of time to actually reach all of the parts here. Yeah. I also wonder when it's actually going to start to make a pupa. I don't know. So stereo. Here it is. Here it is. See? Happy and moving around. Yeah. So it molted already, I think, 3 times or so. Yeah. Yeah. Google says it can take a couple of months, up to 2 years. Wow. Yeah. It's not able to escape. It's not able to crawl up the smooth. Yeah. Yeah. So let's put the lid on top again. Let's turn off the light. I think it likes to prefer to be in darkness. And here we are again. Okay. So yeah. So what I might do actually, or I'm going to save this maybe in a couple of, maybe later I'm gonna put some more of this under the microscope. But I love this. I love those bristles here. I'm a little bit afraid now to actually go up with the magnification, right? Because there is so much mounting medium now on the side. If I go up higher, then there is indeed the risk that I might touch one of the, that one of the objectives actually might touch the mounting medium. I definitely don't want this to happen. I'm talking from experience here. Yeah. But really interesting stuff here. Yeah. Yeah, October. I mean, I found the guy in October. So yeah, I don't know when. Yeah, how long he's been living. Yeah. So, but it is a very nice specimen. What time is it? Yeah. So what I'm gonna do now is I'm going to move on a little bit. You know what? This over here has been now. I'm going to check those. Some of you call this tiny little spiders in the urine. I don't know if they. So I'm gonna put this in here. It's been a request to have a look if they're. If I only were able to find them. It doesn't look very much different. Maybe I had go up a little bit with the magnification here. Yeah. It doesn't look so much different. But look, the stain seems to have reacted. Interesting. These streaks. Ah, look here on the side. Yeah, these seem to be. You see, that's now the side of the cover glass. These seem to be indeed crystals that have formed there, where basically the liquid is evaporating. And the pink, yeah. Ah, that's an interesting thing. I have a suspicion. Okay, look. Look. Do you see those things in here, right? And do you see that there's this bright thing around it? It almost looks like maybe these are actually could it be maybe that's the crystal violet that started to crystallize out? Yeah, and maybe because it started to crystallize, therefore it absorbed kind of the stain which was around it. Yeah, over here the crystal violet over here started to crystallize, and that's why we have what we have in here, and therefore it's not stained in here. I think. That could be the reason. So these crystals here might actually be artifacts. They might not, I guess, might not be actually in the urine, but might be actually the crystal violet itself that has started to crystallize. Yes, yes. Here is. Look, I've got the thing here. Where is this? Yeah. Crystal violet, that's the official name. It's a very, very intensive stain. I've before that I've used also methylene blue, which also works quite well. Yeah, yeah. So, okay. So what I'm going to do now is the following. Yeah, I did not rinse the stain. I simply applied it and then yeah. Ah, yeah. Some questions in case I forgot some of them. Thank you. Okay. So, what I'm going to do now is the following. Let me look at the time. It's almost 1 hour. I do want to put something else under the microscope today, which I have not done now for quite some time. So I'm going to move on a little bit. So let's clean up here. I have to show you a pretty sad looking terrarium that I made some time ago. And I say sad looking because it looks pretty, yeah, not so nice. Maybe, I don't know if you look, let me maybe show it. Do you remember this thing here? I added some rocks a couple of months ago, some moss, a little decomposing piece of wood, and it doesn't look nice. Yeah, looked very pretty before, almost like I don't know, a little like a landscape, but not nice at all. And just show it to you here. Let's turn on the light here. This is this was all green on the side. Here was moss, but there is a little piece of wood over here, and now it looks pretty bad. And I think every time when something looks pretty bad, it's actually a sign that there might be something interesting growing on here, hopefully. And this is what I would like to find out. Okay, so what I need to do is I need to take a small sample here. And I want to put it on the microscope slide. And I just realized I need another beaker. You know what I'm going to do? I'm going to do it the easy way. Where is it? I'm going to put it on here. I'm going to, you know what? I'm going to use this. So I'm going to pour away the urea to rinse this a little bit. So, and let's do the following. Let's now use some tweezers and get some of this stuff from the wood on here. This is horrible looking, and that's a good thing. So let's add a little bit of water. This is way too much. Put this away. Let's add a little bit more water. So I'm going to show you now a little trick. If there is so much solid material on here, if I drop a cover glass directly on it, then there is the problem that it's not going to be very flat. So I'm going to now use a cover glass and I'm going to do the following. I'm going to actually wipe everything away here. Okay. Go back a little bit and do something like this. Okay, so yeah, most of the solid material is here on the side, and I hope that this one over here there will be something interesting in here. So let's have a look. I'm going to put away the thing again. I have to. Here is the slide with the thing on it. I have to put it somewhere where I don't lose it. And so, okay. This is how it looks like. Lots of debris. I'm not seeing, normally you would tend to see some nematode worms or something interesting. But there is not really a lot to be seen here. Ah, of course here. Yeah, maybe I wiped it away. Here, plenty of worms. Yeah, okay. So this is basically not under the cover glass, but this is basically there where I, yeah. So they were evidently too big. And here we're in the cover, under the cover glass. Oh yeah, what is this here? A rotifer, I suppose. Yeah. So, careful again here. Yeah. Yeah, so in other words, if you don't have a water sample, just add a little bit of water to some wood and wait a couple of weeks. Yeah, and then you're able to see stuff like this. Yeah. Every now and then I'm just checking the comments section. Yeah, so this maybe was a little bit too extreme of what I've done, is I've wiped too much of this stuff away. I actually hope to be able to see some amoeba as well, because they can generally also be fun. Ah, that's the shell of an amoeba. It's referred to as so-called amoeba tests, testate amoeba. This round thing that you see over here, it's like a the shell of an amoeba, so to say. Acella, yes. What are those things moving? Well, the things that you see, that's streaming, that is simply the that's a nematode worm. That's basically simply the liquid starting to spread. Yeah. Here's another one in the center. The amoeba, they do move, but they move fairly slowly. That's why they're easily overlooked. So for this reason, you have to be a little bit patient. But I'm quite sure that there are plenty of them in here, but maybe it's just difficult to see them. Yeah, here, another one. So, but let's go back over here under the cover glass, because here we can actually go up a little bit more with the magnification. But here, look, there is maybe this one over here. Yeah. One of the interesting things about whatever it is that you're observing, be it decomposing wood or water samples, is if you wait over several days or weeks, you can actually see that the composition changes. So certain organisms start to become more, others start to disappear. But honestly, slightly mixed feelings about that. There could be more interesting specimens. You know what I'm going to do now is I'm going to try something different. I'm not yet going to give up. New slide. Again, some water. And we're now going to do it like this. Here. Something like this. And cover glass on top. Yep. So let's give this one here a try. Yeah. Of course, if you do something like this, then the things are more spread apart. It's more difficult to find them, but when you find them, they're easy to observe. It's a little bit of a trade-off. But generally, I have to tell you, I have seen more interesting specimens before. Bacteria in phase contrast, yes, I can do that, and I'm quite sure that there are plenty of bacteria present here as well. Okay, so let's do the following. Let's go then to phase contrast. Okay. And yeah, I mean, it's many of these tiny dots that you see in the background, these are all bacteria. But sometimes it's difficult to distinguish them from other small things that are there. But maybe in a regular water sample where there is something decomposing, sometimes the concentration is higher. But in general, where is this? Those tiny dots that you see in the background, these are in many cases bacteria. Or see, sometimes there's something swimming around, like in the top here. Yeah, that could be one. Yeah. Bacteria are yeah, fairly small, obviously, and they appear to be dark in phase contrast. So this one that you see moving around there right now, next to the arrow, that's most likely one. Yeah. So, you're surprised that there's so few bacteria. What I assume is that there are probably more fungi growing on there. It could be that the nematode worms and so on, that they eat many of them, and it could also be that they're simply difficult to see right now. Because yeah, normally if you have a real bacterial colonies growing somewhere, then the density is extremely high. But those tiny things that you see in the back, these are all, yeah, mostly bacteria. Those tiny dots. Yeah. So, but indeed, bacteria belong to the slightly more difficult specimens. So I don't know. I could try. You know what? I'm just as a last try. What time is it? Okay, in a few minutes I'm gonna stop again. So, but I want to still give it a last try. And then maybe I'm going to quickly go through some of the questions that you've written here in case I did not answer them yet. So I'm going to do the following. Look. Just dip it on here. The rest goes back. Cover glass on top. This is going to be a very dense. Yeah, but there might be here on the side here. Yeah, I'm going to check that. Over here there might be some sand grains in here, which might which prevent the cover glass from being very flat. So I'm going to try it. Yeah. Again, here it's a little too dense. Let's have a look here. Yeah, I have to tell you, not super interesting. A little bit disappointing for me, but I'm sure there. Here's something. Yeah, yet another rotifer. You see them all over the place. Yeah. And I'm going to try this one now. Again The shell of an amoeba. Yeah, here you basically here now see more bacteria in the background. Here it's better visible. All those tiny dots in the background here. Yeah, these are most certainly bacteria. Yeah. So I'm going to quickly do now the following. I'm going to leave this here and I'm going to quickly go through some of the questions in case I did not answer them yet. Yeah, what does pure urea smell like? Like nothing. Okay. The question of the stereo zoom. Yeah, it would be interesting to know all of the samples your friend sent you. Maybe a topic for later. Okay, thank you. I'll keep that in mind. I'm going to go through all of the samples again. Okay. To make bigger crystals, the fluid needs to crystallize over a longer cooling period. Yes. Red blood cells in the urine. At least the things that we've seen there were too small for red blood cells. So I think these might be other possible cell fragments or other things, but not whole red blood cells. Apparently blood in urine is actually not a good sign anyway. Yeah. Ever set up a screen using different concentrations and conditions, for example, temperature, humidity, maybe you can optimize the crystals to grow better. Yes, that's indeed a possibility. One could try to, for example, also try to grow crystals in a vacuum chamber at low temperature. This would also be a possibility, or to warm it up to different temperatures. There are different possibilities to try out, and they will grow to crystals differently. And also, of course, the concentration is very important, because sometimes when you're using a lot of sample, then sometimes the crystals will grow very thick and then maybe not even as nicely visible because they're very thick and dark. And so the amount of substance also plays a pretty important role, right? So yeah, here we actually see more bacteria again. Yep. So it could be maybe that we could not see it before because there was too much movement going on. Yeah. Yeah. Fungi, yeah, could be, I don't know if this could be, let me move this one over here. Yeah, either, I don't know if this is one. Yeah, fungi, this is, I'm quite sure that there are plenty of them here, but they might be difficult to see. Isn't it old, dead, and destroyed red blood cells that make urine the yellow color? The yellow color from the urine is, let's put it this way, the old, destroyed red blood cells, they make feces dark. That's the bile, which is then produced by the liver. Yeah. So indeed, the bile by the liver is actually the products formed by broken down red blood cells. Yeah. So plenty of bacteria here. It could be, I don't know, it could be that those things here where the arrow is pointing to, that these maybe are fungi because these cells are much larger. Yeah. So what we have, yeah, bilirubin. Yes, that's the yeah. Also, for example, if you've got a liver problem, then your face becomes yellow. Yeah, so that's yeah. Yeah. So here we have actually the bile is, well, both have to do with the liver. The bile is actually the thing secreted over the gallbladder, which is produced by the liver. And that is actually an end product of the of red blood cells. Yeah. So look at the bacteria here. There are plenty of them here. Urobilin, also known as urochrome, which is produced when the body breaks down old. Ah, okay, good. Interesting. I know that bile. I didn't know that this is also the same for urobilin. I knew that it's for the bile in the liver is actually formed by the breakdown of hemoglobin. Okay, thank you for researching. So, yeah, plenty of bacteria here. Okay, blood, bilirubin, urobilin into urine. So that is basically some wood under the microscope and microorganisms all over the place, but mostly decomposers, as we obviously, and mostly bacteria. Oh, this one here is wiggling around a little bit. So I think what I'm going to do now, slowly, it's 1 hour 15, and I slowly want to call it a day again. Yep. Yeah, enough bacteria, enough urine crystals and so on for today. I think I'm just going to call it a day today. Yeah, plenty of things to explore. I would like to thank you all for not only for being here, but also for your suggestions, for putting things under the microscope. It's always also a very learning process for me as well, to try new things that I did not try before. I think I'm just going to wish you a nice day, a nice week, as always, and I hope to see you again. And for next week, I'll think of something else again. And okay, I missed a brief question in the chat. I don't know which one it is. Okay, but yeah, if you ask it next week, then it's fine. Okay, all the best, and bye bye, and hope to see you again. Bye bye.